Hi, 안녕하십니까? My name is Julius Sugue from the Philippines. I'm pursuing my master's degree right now at Konkuk University. And I'm happy to share with you today the secret or our activity, how we produce rice without using or without using commercial fertilizer or synthetic pesticide. So this undertaking is under the guidance of my advisors, Professor Dovan Kim and Professor Yang So-Kun. So yesterday we are lucky, we witnessed the global trend of crop production, the challenges that threatening our crops right now. So yesterday Professor Ju has presented these challenges. Similar challenges falls through with our rice production. The big challenge is food security. We can ask to ourselves, can we cope with the increasing demand? Because right now the population worldwide is still growing. So most of the developing countries, rice is their staple food. So can we cope with the demand and supply gap? Then another problem is the pollution or the problem created by the intensified farming technologies, because most of the government intensified the production of rice so that they can meet the increasing demand of their population. But what is happening now? The problem was created, the modern technology created another problem, particularly in the environment. Right now we are experiencing the undeniable threat of global warming or the climate change, which is rice is the most threatened crops. Then something we should do, something should be done to address these challenges and to reverse the current trend leading to sustainable rice production. This can be achieved by environmentally friendly crop production system. It is high time to get back to the fundamentals of agriculture, which is called the sustainable agriculture. The key is to build a sound and scientific principle that will increase again or restore the fertility of the soil, that will lead to a more robust or more resistant to pests, rice that is resistant to pests, and most of all, a good or quality produce. So we simply define or we simply describe natural farming, natural science farming as the farming that respects nature, because it is environmentally friendly, it is sustainable without using any synthetic or any inputs that will degrade the environment. So this amino acid is a nutrient used in the technology of natural science farming. This experiment is anchored in totality about the we will prove if natural farming really can produce. Can we use the technology in producing rice grain? So we choose this amino acid because this amino acid contains protein nitrogen. We all know that nitrogen is the most important nutrient element needed by crops, needed by rice to produce more tillers during vegetative stage and to produce more grains during the panicle initiation or during the panicle initiation stage. That's why we use this amino acid as the nitrogen source of our rice crop. So it is also interesting to know that it can promote protein, it can promote microbe action in the soil, and it can promote nutrient uptake and chlorophyll production. If they work together, it will become beneficial to our rice plant. So how can we make this amino acid? So, I know that we have all the experts here in the natural science farming. So I think there are a lot of we have common standards, or there are some differences because we need to consider some climatic condition. We have different climatic conditions in different countries. So anyway, the fermentation might be the different fermentation process or the duration of fermentation. But the most I want to emphasize here the common ingredients, which is very basic, which is very readily available in all our in the household. We just need the fish byproduct, brown sugar, ISO, porous paper or mosquito net, rubber band, and clay or jar or the container. Then the process of making fish amino acid is very simple. Just chop into pieces, small pieces, the fish byproduct, and mix with equal amount of sugar. Mix well, put into the container. Then seal it with sugar again. Then cover with porous paper. Then put in a well-ventilated, dark, cool place, so that we will wait for the mixture to ferment. It will take around 2 to 6 months so that we can produce a good concoction. And it can be used by foliar or by soil drenching. We can water directly applied into the soil or through mist spray in the foliage of our crops. So the experiment was conducted in Maju, 1.5 hours from Seoul by car, and we conducted transplanting last May 27, 2016. And the rice we use is the Japonica type rice. It is common in Korea. The local term for this rice is chuncheong rice, and it has a 150 days maturity. We employ the randomized complete block design with 4 replications. Each plot measures 5 by 5, or the total experimental plot is 400 square meter. Then the fish amino acid treatment, for our control, we did not use any fish amino acid. For treatment 1, we use the 2000 dilution. For treatment 2, we use 1000 dilution, and for treatment 3, we use 500 dilution. These are the nutrient. We follow the standard or the book or the references we have gathered from our expert. So type 2 treatment was applied during the active tillering stage. Anyway, the component of type 2 nutrients are these: OSN or Oriental Herbal Nutrient, FPJ or fermented plant juice, PRB or brown rice vinegar, calcose, and lactic acid bacteria, and also the FAA. Depending on our treatment, on the dilution of the experiment. So we applied all the nutrient during the 13 days after transplanting until the 14 days after transplanting. This period are the vegetative stages of the rice crop. We applied 5 times with this nutrient. Then for the next treatment, we use the transition treatment for 2 times. During the panicle initiation stage. Panicle initiation stage can be determined when you get a stem of the rice, you can see a 2 millimeter white particle in the stem or inside the vascular membrane or the xylem. So you can see a 2 millimeter white, protruding white, small piece of white membrane. So we determine that when we see that during after 59 days after transplanting. So we spread immediately with this transition treatment. Then we repeated the application during the heading stage for 80 days after transplanting. The components are these. Then for the next treatment, we use the type 3 treatment during the flowering stage. So it is visible when you see the rice panicle already appearing after the heading stage. You can see the spikelets are now producing white pollen. So we already sprayed with this treatment containing all the OSM, almost similar component, but they differ only on the dilution rate or the concentration. Then last treatment we applied in the rice crop is type 4 treatment during the milking or soft dough stage. Then these are the activities we have done before producing a good grade. So before transplanting, we scattered or we broadcast IMO4 in the soil. Then we use a mechanical transplanter because in Korea they are already fully mechanized. Manual labor is not common in Korea. But in the Philippines we are still using manual transplanting. But here we use this machine. So the planting distance is 30 by 15. It is already predetermined, or the setting of the machine is already this way. But if you will use manual transplanting, you can manipulate the distance. In the Philippines, we use 20 by 20 or 20 by 15. But here in Korea, they are using 30 by 15. Then the average seedling per hill, the number of these per crop, it's quite many. It is 10. Usually, in the Philippines, we use 1 or 2 to 3. But here, because the machine is already okay. Then with control, we use this. We do manual weeding. We release snail, and we allow propagation of proliferation of snail because somehow they can help us control the weeds, and we keep good water level to suppress the weeds. Then we do replanting. When we observe that there are dead hills, then spraying of all the nutrient. We apply the nutrient through foliar spraying. We use also liquid IMO and bioinsecticide when we observe that there are pest-ridden plants. Then these are the transition or growth transition of the crop. You can see from weak seedling to this very robust and productive rice plant. Then you can see the root profile. This is the control and this is the treatment. You can see the profuse and the sturdiness of the roots and the thickness of the root when they are applied with this amino acid. Then you can see we just took this picture just to compare the resistance to lodging or to resistance to strong winds. Natural grown crop is more stronger than the conventional. This field is around our field. So you can see they are already lodging. Then these are the results. You can see there is a significant improvement in the growth parameters of rice, particularly on tillering, on root length, on plant height, number of plants per hill, root length, root volume, leaf area index, and plant height. So you can see control has the least figure, and treatment 3 exhibited the highest performance compared to all other treatments. Then in this figure, in this table, I want to emphasize here that number of particles is the most important component to determine rice yield. So you can see there is a significant contribution of this amino acid on the number of particles per hill. Then to summarize my report, I can say that FAA significantly improved rice growth. Use of FAA with 500 dilution holds potentiality to boost growth and productivity of rice. Or right now, natural science farming will produce high output with natural inputs. So I want to end my report by simply shouting, natural science in rice, we can do it. Thank you.